So I hope you all can see my presentation, Transcending Protein Expression, Next-Gen In Situ Proximity Ligation, and the Advent of Spatial Interactomics. So just a brief slide about Navinci. So we're a company in Sweden. We were launched in 2020. We are using distributor markets, and we have a global presence. And we have developed products for proximity ligation assays since the very start. If we go in and take a look at spatial biology and the different technologies and its uses, and a little bit of the history of spatial biology, we have, on the one hand, we have transcriptomics, which is widely used. It's give customers and users very well picture of the spatial context. It's, however, not focusing on proteins, and you have no possibility to look how the protein actually function, how they interact with each other, or how they phosphorylate, or what and when they are actually active. Looking at RNA, you get a one-time picture. This is how it looked at this time. It's not, this is how they work together. Going in the field of proteomics, then, where you actually look at the proteins, and you use multiplex IF technologies, where you can look at many proteins at the same time, looking at the protein expression, but you still have no real possibility to see how they function. To do that, you need to go into the field of interactomics. And this is where our method, our technology, and our products come in, giving you the spatial context, the protein expression, but also the protein function. So if we kind of point out the needs and the technology challenges within this field, first we have a need of sensitivity and specificity, so that you are being able to have a strong signal enough to be able to see low abundant proteins, and you need to have the possibility to look at them with very high specificity. You need to be able to see them without having problem with background and non-specific signals. With resolution, there is a big need to be able to see single events, look at single proteins, single interactions. There is a more and more demand of higher throughput to be able to run many samples at the same time. And in the last few years, the multiplexing has become rather a wanted need, and the usage of multiplexing has been shown lately. And when you're looking at the very complex data set of everything from interactions in cell and tissue and multiple products, interactions and protein expressions, you need a possibility to do data interpretation as well. So the standard method that we have done for many years to look at protein interactions is to use a normal immunofluorescence method, and you look at two proteins, two different signals, and when they overlap, you say that you have an interaction. The problem with this is that the diffraction limit of a normal microscope is around 250 nanometers, meaning that everything within 200, closer to each other than 250 nanometers, will appear to be a protein interaction. While the size of protein interaction is rather 40 to 50 nanometers, which means that it's a good span of false positives. So it's a high risk of misleading to use this method. And this is where our methods in situ proximity ligation comes in, and it allows the user to look at protein interactions, not only in between cells, also within cells, within the nucleus and within the cytoplasm. And this is a quick overview of the technology. So the technology is based on dual antibody recognition, meaning that we have two binders, two primary antibodies that bind to their respective proteins in this case. And the next step is to add secondary antibodies connected to DNA oligos, which we call Navinci. When the two primary antibodies are within 40 nanometers from each other, the oligos on the Navinci will be able to connect and create a DNA circle. The DNA circle can then be amplified to a single-stranded long DNA strand with repeated sequences that we could target with fluorescent label oligos, making every proximity event a very distinct, a very strong channel with a high amplification that could easily be visualized in a fluorescence or light field microscope, and then quantify by everything from just counting signals up to more advanced data analysis technologies. And this is a comparison of how an interaction, when you're looking at two overlapping IF signals on one single cell, looks with a very high magnification, of course. With the same magnification, you see to the right, you see the PLA signal, and it's very easy to see that the possibility to count, to detect, and to visualize the single interactions. So the uses of the Navinci technology is you can look at protein-protein interaction, as I showed in the image, but you can also look at the post-translational modifications such as phosphorylations, where you have one antibody directed to phosphosite and the other antibody directed to the protein, creating a very specific assay for the phosphorylated protein. You could also direct the antibodies to the same protein and have a very strong single protein technology, and to see where the protein is localized. This is especially important for low abundant proteins. And it's been more and more widely used for antibody validation, where they have seen the PLA superior to IHC in validating primary antibodies. And the needs is of course to understand biology, to evaluate drug targets, understand response to treatment, to use the technology for patient stratification, to understand disease progression, and to validate predictive and prognostics biomarker. I will just briefly mention how our product portfolio is divided. We have one part of the portfolio is built up by flexible kits, where the customer and user could use their own primary antibodies on their own tissue completely open to the user. We have some target-specific assays where we have already chosen the primary antibodies. We have already optimized the primary antibodies, so the assay is optimized and ready to go. And we have also developed automated kits that is both flexible and target-specific for run on LicaBond RX. The most common uses of the PLA technology is so far the flexible possibility. And there we have developed and divided the two materials that is most widely used, cells and tissue, because they are very unlike each other. It's very different to look at protein interactions in a cell and in a tissue, where the tissue is more complex and it's more need for background reduction, and it's more need for clarity, and it's a little bit more complex system. In basic, while cells are easier, and we have a short protocol, simplest analysis, and it's of course used for the same uses as always. And for the other hand, we have the Navinci tissue that visualize signals very clearly that would otherwise be obscured by tissue background. And using the in situ PLA on tissues, and you can analyze protein interplay within the intact tissue morphology. This is an example from a study that was published a few years ago, where a group in Vienna, led by Nicole Heinzel, showed that using the Navinci cell product, they could unveil that p53 aggregate could be a potential independent prognostic marker in ovarian cancer. And they argued that using PLA to verify ELISA results is an additional promising diagnostic marker tool that allows the analysis of FFPE samples. So it's a little bit a proof of what we're saying, that it actually have promising diagnostics benefits and uses. The flexible kits are available both for use in fluorescence in a fluorescent microscope, but also chromogenic in a readout in light microscope. It's all dependent on the user's choice. We have a third method within the flexible world. Well, you can still use your own antibody, but with the triflex cell, which is a new addition to the flexible catalog, you have the possibility to not only look at the interaction between two proteins, but you also will see them, how they look while they are unbound from each other. So you will have the possibility to visualize and quantify three spectral distinct fluorescent data points, and you can quantify the level of how many, what is the ratio, sorry, of expression of protein A, how many of those are actually interacting with protein B, and the same wise, likewise with the protein B, which is taking the next step in looking at the protein interplay to be able to actually see where they are when they're unbound, where they are when they are bound to each other, and calculate the ratio of it. If we go into the target-specific world, we have chosen to start with the immune oncology, because that is very, very widely used within the spatial biology field. And within the immune oncology, the PD-1 and the PD-L1 is the clear choice of biomarker that has been very, very well used and very well studied for a long time, mainly by looking at the expression of only one of the two markers. But with the PLA technology, you will now, of course, have the possibility to look at the PD-1 and PD-L1 interaction. And the PD-1, PD-L1 interaction is a very common biomarker within immune oncology, and it's most studied because its ability to inhibit T cell activation, allowing tumors to evade the immune system. And the PD-1, PD-L1 has the ability to predict the efficiency of immune checkpoint inhibitors, and therefore this interaction helps identify patients who are more likely to respond to immune checkpoint treatment. And this is shown in another research that have been published very recently, actually, It's been done by Professor Patrick Micke and Dr. Karina Strell at Uppsala University and the University of Bergen in Norway. And in this study, they looked at the cohort of 305 retrospectively early stage non-small lung cancer patients. And they detected and they looked at the expression of PD-1, PD-L1, and they looked at the expression of PD-1 by itself using IHC, and the same thing with the PD-L1. And interestingly, what was very interesting with their study is that only a percentage of the people that expressed both PD-1 and PD-L1 actually had the interaction, around 54%, or actually 54% of the patients that had the expression of both had the interaction, which indicates that is it really possible to say that a patient do express the PD-1, PD-L1 interaction and will respond to treatment by only looking at either one of the two biomarkers. And they took this one step further and looked at the patients, and they looked at if they could select those patients by the expression of PD-L1 by itself, and see if they could separate those that had benefited from immune checkpoint treatment and those that had not benefited on checkpoint treatment, and they could actually see no difference. But when they looked at the same patient cohort and looked at those that had a high PLA score and a low PLA score, they could actually significantly separate those that had responded to the treatment and those that hadn't responded to treatment. So they argue that looking at PLA prior to selecting patients will be much more efficient, so that the right patients get the right treatment. But it's no need to stop at the PD-1, PD-L1 using the PLA technology. You could go with the flexible product. You can look at the phosphorylated PD-1, which is very crucial since it acts as a marker for T cells that have been suppressed by the PD-1, yeah, interacting, indicating a state of immune evasion, exhaustions within the tumor microenvironment. You could also look at downstream protein interactions, such as the PD-1 and SHP2, which directly impacts the T cell activation and thereby the effectiveness of PD-1 cancer immunotherapy. So it's no need to stop at the target-specific product and stop at one interaction. Using the PLA, you could go even further and go look at phosphorylation and downstream effects. And here we have a professor at the University of Queensland, Associate Professor Arutha Kulasingi, and he has used the Navinci PD-1, PD-L1 product and integrated it with PhenoCycler Fusion to look at how you can see how you can study the effect of PD-1, PD-L1 within macrophage and T cell enriched neighborhoods in immunotherapy resistant tumors. And what they could see with this study was that across different patients' response, A, complete responders, B, partial responders, C, those with stable disease, and D, those that didn't respond to treatment. Arutha and his team were able to compare neighborhoods which were PLA positive and those that were PLA negative. Cell typing and cellular neighborhoods, cell typing of cellular neighborhoods across response groups. They compared the cellular neighborhoods with and without including in situ PLA positive clusters, and showed that different PLA scores affected the neighborhood distribution across the different responder groups. Our second target-specific assay is the CD8-MHC1. And looking at the CD8-MHC1, you could get an increased understanding of this signaling pathway. It's also a very used marker to look at the activation of CD8 positive T cells. And it can be used for evaluating the immune checkpoint inhibitors and looking at the complete immune response on a higher scale. And if we take a look at the last protein product category, which is the bond, the automated products, which is of course to have an automated protocol, you increase your throughput, you increase the robustness, and you increase the reproducibility, not only between experiments, which is already very high, but also between patients and between laboratories. And using the bond on the Navinci products on the bond, you can analyze up to 150 samples per week with one instrument and one person. And the Navinci bond automated products do everything from bake and de-wax the sample through antigen retrieval and the full proximity ligation assay. So the entire workflow is done by the machine. And we have validated this on many assays and on many tissues. And this is just 3 examples of PD-1, PD-L1, CD8-MHC1, and also SAPLAT interaction. And one thing that we have been working hard on the last year, which we think is very important for not only for us, but also for the users, is that the possibility to integrate the in situ PLA within other platforms, such as Akoya's platforms, Bruker Spatial, and molecular instruments to look at the RNA protein binding. And this is an example where Dr. Charlotte Stadler from KTH in Stockholm, here in Sweden, did the PLA on a Leica bond and then transferred the slides into the PhenoCycler Fusion and run the multiplex panels. So combining the 2 technologies, but still fully automated. And they used it to see if they could correlate the score, the PLA score, with other markers and compare it between complete responders and non-responders in bladder cancer patients. And they demonstrate that increased number of interaction corresponded with the immuno checkpoint treatment response. Here's another example done by Arne Christiansen that works with Bruker Spatial. So he did the technology on the Cellscape machine. And he also had interesting data regarding the PD-1, PD-L1, and the usage of looking at the interaction in selecting patients, because he, to the left here, you can see that, what is shown here is on a colorectal carcinoma tissue, The patients were selected by those that had that didn't have an interaction expression of PD-1 and PD-L1 in the cancer, but they could find it in the stroma within the T cells. And the interaction between the PD-1 and PD-L1 was also seen within the stroma, but not in the tumor. This would indicate that these patients would actually benefit from the immune checkpoint inhibitor treatment, but they would never be selected if you looked only on the PD-1 and PD-L1 expression. And on the other side, we have an example, or they had an example where they looked at lung carcinoma and they looked at PD-L1 positive lung cancer samples. But they saw no PD-1, PD-L1 interactions in these patients. And based on the standard stratification, these patients would have been chosen for immune checkpoint inhibitor treatment, but they would likely not benefit from it. And here is a short example of how it looks when you combine the in situ PLA with an RNA readout. The assay is completely automated on the Leica bond. And the application is, of course, were used in this study to assessing the downstream effects of cell signaling. And now, finally, I will go into the latest innovation of in situ PLA, the latest addition to our products, and that is completely new, and we're completely alone with being able to offer this, and it is a multiplex of PLA. So you get multiple in situ PLA detections in the same sample, in the same run. And in the Navinci product that we have out, you see 3 different targets in the same product, same run, same tissue. And the assays that we have chosen in the first panel is the PD-1, PD-L1, and the CD3, which is a T cell marker, and the CD8 and MHC1. And combining these, you could understand how the immune evasion mechanism by looking at immune evasion with the PD-1, PD-L1, and how it correlates to the T cell activation with the CD8 MHC1. But you can also go one step further and use the panel to predict therapeutic outcomes and guide immune therapy strategies to improve treatment if you also add the benefit of the T cell. You can measure the level of T cells that actually are activated via the CD8 MHC1 pathway, and how they together work with the PD-1, PD-L1 for immune evasion. And we have coming up very soon a new panel that will be done and that will be available, where we have, I can see it's misspelling here. We have the PD-1, PD-L1 interaction. That is our star. You might have noticed that. And we also have the PD-1, PD-L2 interaction, which is an added benefit, because then you could actually see through which pathway patients and immune evasion is done. So it can also be done through the PD-1, PD-L2 binding. And in this panel, we will also add the PD-1, PD-SHP2 that I mentioned earlier. So this panel will be a very powerful way to look at the PD-1, the PD-1 were complete how do you say? I forgot the word. The complete pathway of looking at the PD1, PD-L1, and PD1, PD-L2, and PD-1 SHP2. And just a quick show to see that the Naveniplex has been performed in many different cancers and in many different tissues. And finally, we have something that is not really released yet, but it will be available from the fall. And it's a service that is performed at Navinci here in Sweden, and it is Naveniplex Omni, and it's a 9-plex PLA, where you can look at 9 targets together in one run, and the targets can be selected by the user from a list of 50 different proteins that could be combined to a panel of either single proteins or of interactions. And in this panel here that we show, we have, you can see the PD1, PD-L1, of course. We have a CD4 to look at T cell receptors. We have the CD8 MHC1, the PD1, PD-L2. So you can see that the difference between the interactions between the PD1, PD-L1, and PD1, PD-L2, and why it might be very important to see which pathway and which interaction has been used. We have in this picture as well chosen the beta-catenin and E-cadherin interaction, which is a marker for cell adhesion, which is especially important in cancer. We have the LIC protein to look at the regulation of T cell function. PD1 SHP2 to see the effectiveness of PD1 cancer immunotherapy. We also had a LAT-SAP interaction, which is an initial stage in the T cell receptor signaling, and finally a T cell marker. And this is just one panel that we build as an example. There is a possibility to build a wide variety of panels by combining up to 50 different protein targets. This is just another example of another panel. It's mainly the same, but you also added the CD19 and CD21 to this panel. And with all of these technologies, with the flexible products, with the target-specific products, with the automated products, and with the multiplex through Naveniplex Kit or Naveniplex Omni service, we have been able to address all of the needs and technological challenges. This is the list of the product portfolio that is available through Blossom Bio in Taiwan. And we have the service, the Naveniplex Omni service, that will be available from the fall. Could also be ordered through Blossom, but it's performed in Sweden. So thank you very much for your attention. I hope I managed to keep at least almost in time. And now I'm very happy to answer any questions. I've already got a few questions from Haley that was asked earlier by some users, and maybe I can start with those. And then if you have any questions, just please either write them in the FAQ chat or just ask. So the initial question is, what's the difference between PLA products from Navinci and Sigma, which is the original PLA that was developed from Olink here in Sweden back in 2008. And there is two ways to look at this. If we look at the flexible products, the usage of the technology is the same. The Duolink product line, which is sold by Sigma, have the same usage as our flexible kits, if we do not count the Triflex kit, which is completely new. But the difference is that the Sigma kit was developed in 2006 and the Navinci kit was developed in 2018. So it's 12 years of improvement to the technology. So the signal is stronger, the background is reduced. We also have optimized the kit, one for cell and one for tissue, which is very beneficial for the customer, especially the tissue kit, because frankly, the Sigma kit works, but it doesn't work very good on tissue. If we look at the target-specific kit, the automated kit, the Navinci kit and the Navinci Omniservice, there is actually no need to compare to Sigma, because they do not have any of those in their catalog. Next question is, how do we know the PLA detected signals were indeed coming from protein-protein interaction, and how to design the controls for this purpose? This is a very good question. And in general, what you see with the PLA technology, the Navinci PLA, is you see a proximity between the two Navinci bodies, as I showed earlier in the workflow. So if you have an antibody against one protein and you have another primary antigen against another proteins, they are within 40 nanometers from each other, then they will generate a signal. And the feature that called dual recognition, which is that both antibodies needs to bind to the target at the same time within proximity, strongly reduces the effect of any non-specific binding of either one of the single antibodies, making a signal from a PLA assay much more reliable than a signal from the two single antibodies, just because you don't see the non-specific signals from either one of them by themselves. So you could very well trust the PLA signals, but however, of course, it's a biological process with antibodies and other factors. So it is important to have technical controls to be certain that you see is the actual interaction. And what we recommend as technical controls is that you emit one primary antibody and then the other primary antibody, so that you get two negative controls where you have one primary antibody. And you also could add a control where you have non-primary antibodies. If you do this, you will be able to see that you don't have any binding from the secondary antibodies to each other and to the surface, and that you don't have any unspecific binding of one of the secondaries to the primary antibodies or to the corresponding Navinci body or the other Navinci body, creating an unspecific signals. If you do these three technical controls, you can be very certain what you see is a true proximity between your two antibodies. If you also add the biological controls, such as stimulated or unstimulated cells, or biological important tissues, you gain even more confidence in what you're seeing is the correct sample. But to look at if you have a PLA signal that comes from a proximity of the right primary antibodies, and only that proximity, the technical controls are the one that's needed. And how do we quantify the signals? And yes, of course, in cell samples, it's very easy, because if you have a well optimized cell assay, you get separated signals as distinctive dots that could be counted, and you count them, how many signals per cell, and you compare it with two different experimental settings, such as stimulated, unstimulated, or other ways. So that's for cells. It's very simple. For tissues, it's a little bit harder, because then it could be not as easy to separate the single signals. So there is a couple of ways to quantify signals within tissue, and one way is to look at how many clusters of signal you have per slide, how many positive cells you have, and you could also look at the total fluorescent intensity combined with looking at how many signal clusters you have, to get a very specific quantification. And the fourth question here is, as we know, Navinci offers 2 types of products based on sample time. Can the Navinci cell kit be used for tissue samples? Also, can the tissue kit be used for cell samples? Also very good questions, and the answer is very straightforward. The cell kit cannot be used on tissues, because you will run into background. The tissue kit can be used on cell samples. You could even reduce the protocol for the tissue kit with 1 step and run it on the cell. But the tissue kit, since it's a little bit more complex, it's more expensive. And if you know what you're going to look at, and you know that you're going to look at cell, then of course the Naveniflex cell is the kit for you. But if you want to look at both, the Naveniflex tissue accommodates that. I can see that we have got new questions. One from Eric. Is it possible for Navinci PLA kit to generate false positive signals using just 1 antibody? We have detected such signal using Sigma's PLA kit. And yes, it is possible. And that's one reason why it's needed to calibrate your system very well. And when you get false positive signals using only 1 antibody, what you see is you have 1 antibody, you have the corresponding secondary antibody, those 2 are already there, and you have the free secondary antibody that doesn't have anything to bind to. And in some cases, this secondary antibody will bind to the surface of the tissue or the cells, and will in some cases be able to come in proximity to the other navenibodies, and then creating a PLA circle and PLA signal. Or the secondary antibody could bind to either the primary or the secondary antibody. Even though it shouldn't, this can happen. But that is a very less likely event. So what you do when you have this problem, you try to decrease the antibody concentration of the antibody that's already bound in this technical control. So you decrease this concentration, and then you decrease the risk of having false positives. Another thing that we have seen is very important is to use a preheated washing buffer. So you have the washing buffer in the 37 incubator. You take it out and you use that for the washes after the navenibodies. That strongly reduces the risk of antibody binding to the surface. And this one, and probably the most prominent reason for 1 antibody background, as we call it. So it's possible, but it's also possible to reduce it. And even though the Sigma PLA kit is of course not our kit, but I can still, if you send me a question through Haley Blossom, maybe I will be able to, I can give you some advice how to modify your system.
Okay, if there are any questions, if we don't have further questions for now, maybe we can finish today's webinar. Daniel, thank you for today's talk, and thank you to all the scientists attending this webinar. Regarding this webinar, you can find more information at navinci.com. And if you have further questions, please feel free to contact Boson Bio. We are glad to provide support for you. Thank you, and I hope you have a good rest of the day. Thank you, Daniel.
Yeah, thank you for the opportunity to talk to you all. It was a pleasure.
Okay, thank you. Bye-bye.
Bye-bye.